Thanks, Al. Hi, everyone. My name is Jessica Marr. I'm a group leader and associate professor at the University of Queensland. It is my great pleasure to start this session and co-chair it with my colleague, Professor Woong-Yang Park. As you heard from Al, we're going to be talking about non-infectious disease research and single-cell biology. And it might seem strange to define this session as such, but I think COVID has really forced us to consider the impact of infectious disease research. But still, it's really important not to lose sight of what non-infectious disease studies can tell us, especially when it comes to single-cell biology. So the studies you'll hear about today come from an exciting lineup of speakers who will shed light on what problems we can solve with single-cell research in the non-infectious disease space. So it's my great pleasure to introduce our plenary speaker, who also happens to be our co-chair, Professor Woong-Yang Park. Professor Park is director of the Samsung Genome Institute, where he provides a clinical genomics and bioinformatics platform in the Samsung Medical Center and Sung Kyung Kwan University. Professor Park's research studies the cellular dynamics of tumor microenvironment upon anti-cancer treatments for single-cell and spatial genome analysis techniques. He focuses on gastrointestinal cancer to understand the regulation of tumor immune networks in response to immunotherapy. Professor Park has been integral to our HCA Asia community. He is part of the ADA project that Sham talked about yesterday, and that project is seeking to understand immune diversity in the Asian population. Professor Park has also recently founded a venture company called Gingivo-Buccal Inc., which provides clinical genomic data and an AI platform for biomarker discovery and genomic diagnostics. So that's a huge range of exciting achievements, and we look forward to Professor Park's talk.
Okay, good morning, everyone. Hi, so Woong-Yang, and then from Samsung Genome Institute. Happy to discuss our current research on the single cell. And as introduced by Jesse, and then we are more interested about clinical genomics, especially using the single cell techniques. So today I'm going to share our current achievement related to investigating the tumor environment with the single cell techniques. Here's my complete interest. As you know, the cancer immune system is quite an interesting topic because we know about the cancer immunology well, but still we do not know the exact mechanism or some the cell interaction within the tumor tissue. So the cancer immune cycle presented by Chen and Melman B.A. It's not actually it's natural some phenomena to control the cancer within the patient. So the cancer cells, with the hypoxia or other mechanism, they die and then they release some antigens, and then presented by the antigen-presenting cells, and then the educated T cell and the circulating T cell, they control the tumor growth. So it's a natural some physical mechanism to control the tumor growth, but we can use those mechanisms to control the cancer through the drugs targeting the cancer cells or targeting the immune cells. So maybe we can, if we can understand well about those mechanisms, probably can control the cancer more efficiently through some specific drugs. So, but the problem be not simply the interaction between tumor and the immune cells. So maybe the tumor microenvironment, including small cells, other type of cells, is quite important to control the mechanism for the tumor immune cycle. So based on some pan-cancer the analysis, they published that nice paper related to the subtype the tumor microenvironment based on bulk RNA sequencing data. So they divide into the 4 types. Based on this classification, we can see some clear difference in the patient survival, with the immune enriched and non-fibrotic cases be a better survival overall. kind of some tumor agnostic, so tissue type agnostic pattern. So we can apply this idea to all the cancer types. We are focused on the gastrointestinal cancers, especially the gastric cancer, the Asian, the cancer types. So maybe the nice paper by Patrick Tan's group, and then they published it 200,000 single cell data in Cancer Discovery recently, and then they provide a nice the data related to gastric cancer single cell data. And they especially focus on some microenvironment interaction or maybe some rare cell types relating the gastric cancer. I would more focus on the immunotherapy response in the gastric cancer, especially that we use the immunotherapy for MSI high cases. Because we expect that the response to immunotherapy is better than any other MSS cases. But the problem be in the clinical trial can get the outcome, the average would be 54%. So that means we can improve the patient outcome based on the immunotherapy, but still we miss that half of the outpatient. But we still do not know that why those patients, even though they have the high mutation burden or some MSI high, but they do not respond well to the pembrolizumab or other immunotherapy. So we can we want to know that what is the mechanism for those the tumor resistance and the immunotherapy resistance in the MSI high cases. So our recent publication related to MSI high gastric cancer and then their response to the immunotherapy in the Cancer Discovery. So we enrolled about 19 patients for this study. Most of them MSI, all the MSI high cases, and then PD-L1 positivity, 100% they show that high PD-L1 positivity, and also clinically half of those patients they respond well, and then CR or PR, the complete response or partial response in the best of response. Then the other half would be a stable disease or the progressive disease. So we analyzed it, those samples, the baseline samples and the post-treatment samples to understand the mechanism for recency or some response to the pembrolizumab. So yeah, there's a clinical response to those pembrolizumab. We can see that those patterns. And generally, first we want to know that what is the difference between the responder and non-responder, and in the exome data. So roughly we cannot see any difference between these two groups. The MMR or some other, the mutation pattern is almost same between the responder and non-responder, but we can see some difference in the Wnt gene mutations. It's one of the intrinsic mechanisms for the tumor resistance against the immunotherapy, but it's not so strong. So we want to know that what is the another mechanism for the immunotherapy resistance. Then also in the mutation signatures, we cannot see the difference between two groups. And then when you're looking at the RNA data, we can see some difference between responder and non-responder, especially in the TCR-related signaling genes are upregulating the responders, but compared to the non-responders. The other pathway is not significant. So also we do want to see, check the TCR clonality, and then we do see some difference in the non-responder versus responder. We can see the TCR beta chain repertoire diversity is higher than the responder groups. And then if we divide them into two groups, in the high diversity in the group or the less diverse group, we can see clear difference in patient survival. So one of the features related to other than PD-L1 or other TMB, we can propose that TCR beta or TCR diversity is important for the clinical outcome to the immunotherapy. So in detail, we want to see the immune cell changes in the pre and post samples. So in global cell types, it's hard to see the some difference between these two groups, but as you see, the T cell is more enriched in the responders, while the stromal cells is more enriched in the non-responders. So as we expected, the T cell be infiltrated well in the responder versus the while with the non-responders not. Also in the high some fibrotic response. I mean, the reaction is important for the response to the pembrolizumab. And then we dissect them other immune cells in the myeloid cell, the enriched in the TAM population in the non-responder, while the tissue macrophage is more popular in the responders. And also in the T cell subtypes, we can see the baseline is almost same, but we can see some changes, dynamic changes in the upon pembrolizumab treatment after post-treatment. We take these samples in the 8 weeks after initial treatment of pembrolizumab, that will be after second cycle. So we want to see it's not exactly post-treatment, it's kind of on-treatment samples. We can see some difference between responder and responder in the subpopulation of T cells. Especially the CD8 effector T cells expanded in the responders, while the CD8 exhausted T cells expanded in the non-responders. So, and then also we dissected CD8 exhausted T cells in the at the state level. We can see most of CD8 exhausted T cells in the responder they're more stem-like T cells, while the CD8 exhausted T cell for the non-responders mostly the terminal differential cell. So that means we do see the same number of the T cells, but those T cells, the changes upon the pembrolizumab, they go into the effector T cells or exhausted T cells. And even for the exhausted T cells, we can see some difference between these responder and responders. So we can explain the clinical outcome of those two groups based on these profiles. And then the problem be the on-treatment tumor biopsy is not easy, as expected. It's not easy, and then ethically or clinically it's not feasible to apply those techniques to all the patient. So we want to analyze the peripheral blood to see any difference between the responder and responders. But actually, we can see the proliferating T cell population in the PD-1 positive CD8 positive T cells is increased. I mean, out of PD-1 positive CD8 T cells, and then proliferating population is increased. And while the Treg population, the proliferating cell population decreased. So that is, we cannot see the difference between responder and responders, but it's a general the pattern for the pembrolizumab treatment. And then the ratio of the CD8 and the Treg is almost same, and also the exhaustion is not different between these two groups. But still, we can see some difference in the PD-1 positive T cells and out of the CD8 positive T cells, and the difference between the durable clinical response group and then versus the non-durable groups. So if we can monitor those PD-1 cells in the positive CD8 T cells in the patient peripheral blood, we can monitor that patient outcome with those kinds of the method. And then especially we also checked some state of the exhausted T cells in the peripheral blood. And then we do see the complete response versus partial response versus the stable disease. We do see a higher level in the PD-1 level in the peripheral blood in the CR versus also in the PR group, but less abundant in the PD-1 positive T cell in the ST groups. Also, CCL7 positive T cells increase in the baseline sample of the CR or PR cases, but while those cells not. Then we can monitor those patient outcome with the peripheral blood with the PD-1 positive CD8 cells or CCL7 positive the cells, and through the peripheral blood, the baseline samples. Anyway, if I summarize, so as a kind of some descriptive study, but actually we monitor the post-treatment cases with the single cell, also the RNA-seq or exome data. So if I summarize that the finding, and we can see some association of clinical response to the clinical response in the MSI high gastric cancer. We can see the T cell receptor diversity. Also, we can monitor the PD-1 positive cells or the CCR7 positive T cells in the peripheral blood. Maybe we can use those kind of markers to monitor the patient with the Also, we to see the some expansion of effector T cells in the after treatment be quite important for the clinical response to the pembrolizumab. Also, baseline the TAM be important for the final outcome for the pembrolizumab treatment. But still, so based on those findings, we want to know, we want to change the tumor microenvironment with the other method to provide the more some friendly environment to the immunotherapy before applying the pembrolizumab or PD-1 inhibitors. So as you notice, we use the combined immunotherapy with the chemo or targeted therapy. So based on those clinical trial, the FDA approved that the first line, chemo immunotherapy in the gastric cancer, in the advanced gastric cancers. But still, we want to know the exact mechanism for the chemotherapy can synergize the effect of immunotherapy in the advanced gastric cancer. So they should modulate that tumor microenvironment through the chemotherapy. So we designed that kind of study to understand that the sequential changes in upon the chemotherapies, and then maybe after that we can see some difference in the tumor microenvironment changes in the immunotherapy. So recently our paper is accepted in the cancer discovery, and then that design was like this. We use the chemo naive, the advanced gastric cancer, metastatic gastric cancer. And we enrolled about 5 patients, and then we take the baseline biopsy samples, and then after 2 cycles of Gelox or Gelox plus trastuzumab, and then we take another on-treatment samples together, and then compare any difference of the tumor recruitment based on upon the chemotherapy. So in the previous study, we just want to know the mechanism for the immunotherapy resistance, but in these cases, actually, we want to know that the changes in the tumor microenvironment upon the chemotherapy. So if we can find something interesting related to changes in the tumor microenvironment in the chemotherapy, we can modulate that the design, the immunotherapy, the combination together. So this case is to be we have 5 HER2 positive cases. Also, half of them be PD-1 positive, and and then the half of them, almost about 5 cases, 7 cases, the poor responder, and then non-responder, and there's 5 cases poor responder. Mutation landscape, we cannot see any big difference between pre and post, also responder versus non-responder, also HER2 positive and HER2 negative. So as expected, after just 2 weeks, we cannot see any big difference in the mutational pattern in the DNA level. And also there's no difference in the mutation signatures. And the RNA level, we do see some difference between the responder and responder. Also in the pre-treatment, on-treatment, it's not clear, but actually on-treatment sample, they show the higher anti-tumor immune infiltrates, and then also in the lower in the pro-tumor infiltrates, and then those responses the higher in the HER2 positive cases. Then it's not so clear, but actually we could find those TME subtype was changed from the depleted type to other immune types. And so based on some statistical analysis to find that some genes that related to the clinical, on treatment samples, we can see some difference between these two groups, so pre-treatment and on treatment. As you see, those inflammation, the upregulated in the migration, upregulated T-cell infiltration or other the pathways are upregulated in the on treatment samples, but still it's not so clear for the related to the clinical response. And then so we look in detail about those immune cells and the tumor command cells with the single cell techniques. And if when you're looking at the non-responder cases, we do see there's the stromal cells to be higher in the non-responders, while the NK cells, other cells, indeed higher in the responders. Then HER2 positive and HER2 negative cases, we do not see any difference, but actually we are more focused. We are more interested about those TAM is emission in the non-responders compared to the responders. And also after treatment, on treatment samples, and after the Gelox treatment, we can see decrease in Treg in non-responders. And then CD8 without T cells are a little bit increased. Also, in effector T cells, it decreased in the non-responders. And also, we want to check the cGASting pathway in the tumor cells, and then in the single cell level, we check that expression of those genes in the all the cases that they are upregulated by the Gelox treatment or Gelox plus in the smart treatment. And then also cGAS and then the STING expression was upregulated in the on treatment samples. That means the chemotherapy may induce the expression of the genes. So, but the response be higher in the HER2 positive cases. And then we are more focused on the innate immune, and then to check that what is the difference between two groups, the pre and post, and then at the expression level. And then the myeloid cell, the subtype is not changed in upon the treatment, but when you're looking at the differential expressions, we can see some upregulation of the pro-inflammatory genes, and then downregulation of the anti-inflammatory genes. So when looking in detail, we can see some anti-inflammatory process or some differentiation, and peptide loading be upregulated upon the treatment, and then it's not clear difference between responder and responders. And then also we can see the M1 macrophage, the state changes upon the treatment. So we validate those findings using the codex. So we use the batch samples, the pre-treatment, on treatment. We do see the large infiltration of the CD8 positive cells and also induction of PD-L1 expression in the responders. So we can see some increase of the innate immune macrophages infiltration on the chemotherapy. So we evaluate with the other data set. It's independent data set with the chemotherapy, and we can reproduce those findings related to immune PD-1 expression or TCR signaling upregulated in the on-treatment samples. And then especially in those changes also in the more prominent in this validation data set. And also we can see the tumor microenvironment subtype changes on treatment. So, and also looking detail about each cell types, we can see some difference in the HER2 positive and HER2 negative. So HER2 positive cases, they show the more higher the modulation of those immune cells in the upon the chemotherapy. And then most of those the features are upregulated upon the treatment, post-treatment samples. But as you see, those responses higher in the HER2 positive cases. And then so we want to know some difference in the CD8 positive T cells because maybe we can target those T cells and therefore the immunotherapy after the chemotherapy. And then the number of T cells is not enough to divide into subtypes well in our study, but actually we try to divide into 5 or 6 different the subtypes, and also we want to know some states of the exhausted T cells based on our signatures. Then especially we cannot see any difference between pre and post cases, but we more focus on the non-responders, those cases with the MET positive cases, and then we need to see some in MET negative cases, they are more terminally differentiated user T cells enriched in the pre and post. While the MET wild type responder group, they are more enriched in the progenitor or some transient cells. That means there's some difference between these responders. And especially we are interested about the LAG3 positive CD8 cells and the other checkpoint inhibitor for Treg or the T cells. So we can see the LAG3 positive CD8 cells and the exhausted T cells enriched in in non-responders. So when you're looking at those patterns in by the codex and the multiple immunofluorescence staining, we see those DC yellow ones, the CD11 positive, those DC was disappeared in the upon treatment. Also, the B cells are increased upon the treatment. So DC is decreased and the B cells increased. So maybe also crossing the LAG3 positive cells are enriched in upon the treatment in the non-responders. So one of we can propose that those kind of mechanisms for resistance to the chemotherapy, and still maybe also in that it's not, it may inhibit that response to the immunotherapy in the later. If I summarize that, the findings, and then the responders with the fibrable remodeling, maybe is it for the chemotherapy? But actually, we can propose that those the group is responders, they have favorable the TME remodeling for the immunotherapy. So even chemotherapy responder, maybe we expect that the immunotherapy response is better than any non-responder for the chemotherapy, because the non-responder be a unfavorable tumor TME remodeling. So if I summarize that, maybe I can take some of those kind of things in based on our chemotherapy remodeling study. We could see some favorable response in TME remodeling, the sequencing activation tumor cells, and then M1 macrophage repolarization to present that antigen more in responders. Also T cell recruitment and NK cell increase. But the non-responders, we do see some LAG3 activation or Winter in the tumor intrinsic activation. Also DC also decreased, and also B cell infiltration increased. So maybe it's another good target. The strategy to targeting. To design some chemoimmunotherapy combination therapy for gastric cancer based on this kind of findings. So we can design, for example, the poor response to the chemotherapy, we can design the LAG3, targeting LAG3, the checkpoint inhibitors, or maybe we can mobilize, targeting the Wnt signaling for the to have to initiate more antigen presentation for the poor response to the chemotherapy. So maybe good response for chemotherapy, we can expect that the better response to immunotherapy, while the correspond to chemotherapy, we need to understand the mechanism also, and we can design the other method to bypass that kind of resistant mechanism for immunotherapy. So I want stop here to thank to our members, especially the Jian actually she led a lot of clinical trial and also the interpretation of those cases, and also Bine, she contributed a lot for the interpreting the single cell data with the GT here. I want to stop here. Thank you very much for your attention.
Thank you, Professor Park, for an incredible talk. We have a really short amount of time, so maybe we have time for one very quick question, which is, can you predict the role of mast cells in the responder group?
Sorry, so which cell?
The question is, can you predict the role of mast cells in the responder group?
Okay. Actually, I just no idea about that, the role of mast cells that I interested in in our responders, but actually proportionally is increased. So maybe some one of the innate response, but actually at this moment I do not have any mechanism for that. Sorry. Yeah.
Great, thank you. So there'll be an opportunity to come back to more questions at the end of the session. We'd like to remind everyone to please ask questions throughout the session. And it'll be really just helpful to keep the questions going and stimulate a lot of discussion at the end. So thank you again, Professor Park, for a really exciting and interesting talk. I'd like to introduce our next speaker in the session. We have Dr. Elena Denisenko. Dr. Denisenko is a bioinformatics postdoc at the Systems Biology and Genomics Laboratory in the Forest Lab at the Harry Perkins Institute of Medical Research. Dr. Denisenko completed her PhD at Massey University in New Zealand in 2018. In the last few years, she's been working with single-cell sequencing and spatial transcriptomic data analysis and interpretation. Dr. Denisenko conducts her research within the Western Australian Cancer Single Cell Consortium, which brings together clinicians and cancer researchers across Perth. So the title of Dr. Denisenko's talk is Characterizing Spatial Heterogeneity in Ovarian Cancer.
Right, thank you for the introduction, Jessica. I'm just going to share my slides. All right, so first of all, I would like to thank the organizers for the opportunity to present here our work on spatial heterogeneity in ovarian cancer. We work with high-grade serous ovarian carcinoma, which is the most aggressive and also most common subtype of ovarian cancer. Patients with this disease, they often are resistant or shortly become resistant to chemotherapy, and the prognosis here is quite poor for these patients. Intratumor heterogeneity, that is having different types of cancer cells within the same tumor, has been linked to chemotherapy resistance and also tumor progression. In our work, we are surveying intratumor heterogeneity of high-grade serous ovarian carcinoma using a combination of technologies. It's spatial transcriptomics, single-cell and single-nucleus RNA-seq. And one of our aims is to identify differences between patients with good and poor response to chemotherapy. We are working with samples coming from the interval debulking surgery from patients who already received a few cycles of chemotherapy. And so far, we have profiled 5 samples with single-cell RNA-seq, 4 samples with single-nucleus RNA-seq, and for 12 samples, we did spatial transcriptomics with Visium. For Visium, we used frozen tissues, and this is just a quick slide to remind everyone how Visium works. So basically, the tissue section of interest needs to be mounted onto these Visium capture areas, which have just under 5,000 spots, where each spot has a set of barcoded capture probes attached to it. At this stage of the development, the Visium spots are relatively large, so we do expect to capture several cells per spot. And it all depends on the tissue, apparently. So in some areas, we can capture maybe a handful of cells per spot, whereas if you have more dense tissue areas, we get 10 or perhaps even more cells per spot. So we perform different types of analysis for Visium, and on the following slides, I'll quickly talk about these three. First, we did copy number inference, and this is because ovarian cancer is known to have a lot of copy number variation. For this analysis, we used a tool called InferCNV, and this is a heat map that was generated by the tool for one of our samples. Here you can see all spots coming from that sample. X-axis corresponds to genomic positions. Red color here shows predicted amplifications, and blue color shows predicted deletions. You can also notice here horizontal black lines, and these separate different CNV-based clusters. Based on this inferred CNV signal, we thought that these spots correspond to the tumor, and these spots correspond to the normal area of tissue section. But in addition to this, we also observed some heterogeneity within this tumor area. For example, here is this cluster shown in blue, and apparently it has a bit stronger evidence of deletion of chromosome 4. And there's another example here is this cluster shown in red, which apparently has some stronger evidence of amplification at chromosome 19. Then we decided to map these CNV-based clusters back onto the tissue to see how the spots are organized. And turns out that quite often spots that are co-clustered based on these CNVs, predicted CNVs, they often co-localize in the tissue as well. And here is a good example for this blue cluster, the red cluster, and the yellow cluster. Also, quite reassuringly, CNV, what we predicted to be normal based on the CNV inference, it was the same area that was annotated as normal by the pathologist based on histological image. Then we performed some analysis to just investigate more the CNV-based clusters. And here is an example of what we see when looking at CNV-based cancer clusters. So these are genes that are differentially expressed between those. And the first gene shown here has stronger expression in this blue cluster. This gene has stronger expression in the red cluster. And the third gene is expressed in the middle of the tissue, but if, for instance, if you look at the blue cluster, it's not really present that much. So what's particularly interesting about these three genes is that two of them have been previously implicated in platinum resistance, and the third one has been implicated in platinum sensitivity. That's based on some existing research and existing publications. So potentially these different, so platinum is exactly one of the agents our patients are treated with. And based on this, within one tissue section, we might have different areas that have differential sensitivity to treatment. And our hypothesis here is that these areas might have also different tumor subclones. We are currently working on confirming the inferred CNVs. This is still work in progress very much. We are doing DNA sequencing here, but preliminary, just very, very raw preliminary results. So far, they're quite promising, so we are hopeful here. Next, we wanted to look at spatial distribution of individual cell types, but because, as I mentioned earlier, Visium does not give us single cell resolution, we actually have to predict this. For this, we used our single cell data, and we performed cell type enrichment analysis using a tool called GeoTem. Basically, for each of the Visium spots and for each cell type, it calculates cell type enrichment scores, and we can visualize these scores. And these predictions, they also reflect the heterogeneity. For instance, quite nice example, these two types of immune cells, they infiltrate some areas of our tissue section much more than the others. Finally, I'd like to talk about our chemotherapy response analysis. And here we started by comparing tumor areas in poor responders to tumor areas in good responders, and just looking at the differentially expressed genes. Here we again used our single cell data in order to see what cell types express those genes. So this plot shows genes that were overexpressed in tumor areas of good responders. We refer to them as good response genes, and they are largely specific to cancer-associated fibroblasts and a few types of immune cells, which tells us that tumor areas of good responders are infiltrated with these cell types much more than that of poor responders. In terms of genes that were overexpressed in poor responders, it was quite the opposite. So these genes were more specific to cancer cells, and a few were specific to myofibroblasts as well. We then asked whether any of these genes have association with progression-free survival. And to check this, we used an independent cohort of just under 700 ovarian cancer patients who received the same treatment as our patients. And I found that indeed for some of these genes, the high expression of those genes was associated with shorter progression-free survival. We then investigated treatment response heterogeneity again within individual tissue sections, within individual samples. And I'm going to show you the results of these using one of our good responders as an example. So in this patient, we identified 5 CNV-based cancer clusters that are shown here as blue, red, gray, purple, and brown. And then we looked at the expression of poor response genes and good response genes across these 5 cancer clusters. We again found some heterogeneity, and what you can see at these plots is that blue and red clusters were different. They had lower expression of good response genes and higher expression of poor response genes than the other 3 clusters. So with this, we again think that there is some treatment response heterogeneity happens here, and we have this area that's supposed to have good treatment response, and the red and the blue area, we predict that they have poor response. And again, our hypothesis is that these are different tumor clones. But what's particularly interesting again about this patient is that it's a good responder. But what we know is that even after initially good response, patients often relapse. So if this happens to this patient, it might be these red and blue areas are responsible for that. So just to summarize, we have applied Evasion to post-chemotherapy high-grade serous ovarian carcinoma samples. We compared our poor and good responders, and we also identified different tumor areas that have a range of different properties, and we hypothesized that they are different tumor subclones. At the moment, we are working on confirming those predicted CNVs using DNA sequencing. It is still work in progress. Then we are planning to perform cell communication analysis using a tool called NATME that was developed in our lab recently. And we also hope to expand our study to include larger number of patients and to also include pre-treatment samples, because it would be particularly interesting to know, first of all, whether we have that much heterogeneity before treatment, and secondly, whether we can actually predict response for these patients. And just to conclude, I would like to thank everyone involved in this project. These are the members of the Forest Lab who work on this project, and these are our external collaborators from different universities and hospitals and institutes across Perth. And these are funding bodies who make this research possible. And that's it. Thank you for listening.
Thank you, Elena. Right on time. That's brilliant. That's a really cool talk. I have a question. You started off talking about intra-tumor heterogeneity, but you also highlighted that the Visium technology is not single cell. And so I'm just wondering, how do you reconcile the potential influence of maybe differing cell numbers that contribute to your analysis of heterogeneity?
Yeah, that's definitely a complicated area. So one of the things we're trying to do is to predict cell type distributions. Another thing we tried actually is it was kind of separate arm of this research that I didn't talk about today, but just to briefly tell you about this. So we are actually trying to generate, generating perfectly matching single nucleus data and vision data. So we are just using the adjacent sections, and we are hoping that integrating these two modalities would kind of help us much better to understand what's happening.
Yeah, I see. That's really cool. Okay, so we have time perhaps for one more question from Sham, who says, nice to see spatial distribution of CNV clones. Do the chromosomes that popped up from infer CNV match the commonly amplified or deleted chromosomes in other resources like TCGA?
So we didn't look specifically at TCGA for that. To my understanding, it's still quite lots of diversity in terms of CNVs. So there are no like hot regions that would be amplified or deleted in most of the patients. So it's quite, it varies a lot from patient to patient. And even in our, so we're looking at 8 patients. So even across those patients, there is quite a lot of differences. So yeah.
Yeah, I see. So maybe more to characterize then.
Yeah.
Okay, well, we might end there then, but thank you so much again, Elena, for a really fascinating talk. I'd like to move on to our next speaker and welcome Professor Keisuke Kataoka. Professor Kataoka is Professor of Division of Hematology from the Department of Medicine at Keio University of School of Medicine, and also a Chief of Division of Molecular Oncology at the National Cancer Center Research Institute in Japan. He received his MD and PhD from the University of Tokyo. After serving as a hematologist, he did his postdoctoral work at Kyoto University, working on genetic analysis of lymphomas. At the moment, his team aims to delineate the entire picture of genetic aberrations and clarify the molecular and cellular architecture in lymphomas. And the title of Professor Kataoka's talk is Cellular and Molecular Architecture in HTLV1 Viral Carcinogenesis Revealed by Multi-Omics Single-Cell Analysis. So it sounds really interesting. Thank you, Professor Kataoka. I think you might be on mute. Brilliant, thank you.
最初から。あ、うん。なんか
Do you need me to share your slides?
No, I can. I'm sorry. It takes time, but can you see my slide?
Yes, we can now.
We can.
Address.
スライドショーの再開。あれあ、これだ。まあいいんだ。So it's a great honor for me to have an opportunity to present our recent research. So this is my conflict of interest disclosure. So adult T-cell leukemia lymphoma, or ATL, is an aggressive peripheral T-cell malignancy caused by HCLV1 retrovirus infection. In Japan, the frequency of T-cell lymphoma is higher than in Western countries because of the high incidence of this disease, ATL. Most HCLV1 infections occur in early infancy. The action of viral proteins such as TAX immortalizes CD4 positive T cells, but there is a very long latency before ATL development, suggesting that additional genetic events are required for the immortalized T cells to escape immune surveillance and fully manifest themselves as ATL. To clarify such alterations required for ATL development, we have performed integrated genetic analysis, including whole genome sequencing. Through this analysis, we identified many driver mutations, copy number alterations, structure variations, and related pathways. But before ATL development through the acquisition of such somatic alteration, pre-malignant clonal expansion of HCL-B1 infected cells occurred. Also, HCL-B1 infection and ATL development would be accompanied by substantial changes in non-malignant hematopoietic pool. Here we characterize pre-malignant cells and the multicellular ecosystem in HCL-B1 infection with and without ATL. In this study, using combined single-cell transcriptome, surface marker or antibody-derived tag or ADT, and TCR-BCR repertoire analysis, we analyzed more than 200,000 peripheral blood or PB mononuclear cells from 49 samples, including 34 ATL, 11 HCL-B1 infected carrier, and 4 healthy individual samples. We first performed unbiased clustering all PB cells using transcriptome data and identified 38 distinct cell clusters, including 30 malignant and 8 non-malignant clusters. By using surface marker information, we can easily characterize the cell lineages such as CD4T, CD8T, B, NK, and mild cells with higher sensitivity and specificity than from the messenger RNA information alone. We next focused on CD4 T cells, which contain a large number of HCL-B1 infected and ATL cells. In T-cell malignancies, TCR repertoire analysis is very useful in distinguishing malignant from non-malignant cells. Each colored dot represents an identical TCR chronotype, clearly indicating that these monoclonal populations are malignant tumors. In contrast, black dots are non-clonal TCR chronotype detected cells or non-malignant CD4 T cells. Next, we focused on non-malignant CD4 T cells. Subclustering of non-malignant CD4 T cells revealed 3 distinct clusters. HCL-B1 gene HBG expression clearly separated an HCL-B1 infected T cell cluster from typical naive and effector memory T cell clusters, which showed CD45RA positive and CD45RO positive immune phenotype respectively. By contrast, HCL-B1 infected cluster was characterized by FOXP3 and CADM1 messenger RNA expression, and CD45RO positive and CD25 positive immune phenotype inconsistent with previous reports. Next, we investigated the similarities and differences of messenger RNA and surface protein or ADT expression among uninfected, infected, and malignant clusters in CD4 positive T cells. First, we compared malignant cells with uninfected cells and identified 681 upregulated and 91 downregulated genes in malignant cells. Functional enrichment analysis showed upregulation of various immune-related pathways. Comparison of ADT expression revealed co-upregulation of a wide range of immune-related molecules, including inhibitory and stimulatory molecules, and activation markers in malignant cells. Comparison between non-malignant clusters revealed that MHC class II pathways were enriched in the upregulated genes of infected cells, suggesting that MHC class II upregulation may contribute to HCL-B1 infected cell proliferation by stimulating TT interaction and intracellular signaling. When compared with non-malignant infected cells, genes regulating T-cell adhesion and activation were significantly enriched in the upregulated genes of malignant cells. Moreover, comparison of ADT expression depicted elevated expression of co-stimulatory and co-inhibitory molecules, particularly those related to somatic alterations such as PD-L1 and CD28, suggesting further deregulation of immune modulatory property in malignant cells. Next, we evaluated the TCR repertoires in the non-malignant CD4 positive T cell clusters and found that color-labeled clonal cells were already present, particularly within the HCLB1 infected cell cluster. Importantly, the degree of clonal expansion was significantly correlated with the fraction of HCLB1 infected T cells in HCLB1 carriers. Next, we phenotypically characterized these clonally expanded cells within the HCLB1 infected cluster before malignant transformation. Interestingly, multiple MHC class II pathway genes were modestly reduced in clonal cells relative to non-clonal cells. This reduction was confirmed by a slight decrease of HLA-DR ADT expression, suggesting that HLB1 infected T cells may downregulate excessive MHC class II molecules for pre-malignant clonal expansion, thereby avoiding MHC class II mediated immunity or allergy. HLB1 infection may induce various microenvironmental changes, while additional modulation can occur through ATL leukemogenesis. To uncover the complexity of HCLB1 related to immune system, we evaluated non-malignant populations. Amongst their changes, the most prominent was a marked increase of myeloid cells in ATL, despite a decreasing trend in HCLB1 carriers, pointing to the relevance of myeloid lineage in the ATL microenvironment. We previously identified CD274 or PDL1 structure evaluation, or SVs, truncating the 3' untranslated region or UTL, leading to PDL1 upregulation. To disclose the effect of genetic alterations on the tumor microenvironment, we focused on the CD274 or PDL1 SVs. In this cohort, there were 8 samples harboring CD274 SVs. These samples showed decreased proportion of CD8 positive T cells, particularly effector memory re-expressing CD45RA or EMERA cells. In addition, CD274 SVs restrained clonal expansion of CD8 positive T cells, confirming the immune suppressive effect of CD274 SVs. Interestingly, in patients with CD274 SVs, PDL1 surface marker expression, where a surface protein expression was increased in non-malignant cells, particularly in B and mild cells, despite almost no change in messenger RNA level, suggesting that PDL1 overexpression in tumor cells can upregulate PDL1 level in surrounding cells. As the underlying mechanism of this phenomenon, we hypothesized that overexpressed PDL1 proteins in tumor cells are released into surrounding cells. To test this, we performed PBMC co-culture with Jacquard cells transduced with only GFP or PDL1 GFP fusion protein. We found that PDL1 GFP fusion protein was more efficiently transferred from Jacquard T cells into B and mild cells than control protein, suggesting that the biological property of PDL1 promotes the intercellular protein transfer. In addition, when co-cultured with stimulated CD8 positive T cells, transferred PDL1 proteins suppress the proliferation of the CD8 positive T cells, suggesting that the transferred PDL1 protein is still functional. Taken together, genetically overexpressed PDL1 in tumor cells can serve as a major contributor of PDL1 upregulation in non-malignant hematopoietic pool. So in summary, HCLB1 gene expression clearly defines HCLB1 infected non-malignant CD4 positive T cells. HCLB1 infection and ATL development induced dynamic change in the immune microenvironment, particularly in mild cells. So simultaneous messenger RNA and surface protein assessment can reveal that CD274 or PD-L1 structure variation in tumor cells serves as a major source of PD-L1 in microenvironment, suggesting intricate mechanisms for evading anti-tumor immunity. So thank you. So I finished my presentation.
Thank you. Thank you, Professor Kataoka. That was a really great talk. Do we have any questions from the audience that they'd like to throw into the chat right now? If not, can I ask a question then? In terms of your research, you've highlighted some of the mechanisms where the anti-tumor immunity evasion is occurring. What are some strategies do you think we could use to combat that?
So the one mechanism of immune evasion in ATL is PD-L1, genetic alteration of PD-L1, leading to PD-L1 upregulation. So one option is using immune checkpoint against this disease, and the clinical trial investigating the efficacy of anti-PD-1 antibody is now ongoing. And also, so there are many alterations involving involving molecules associated with immune evasion or immune modulation. So maybe we can use other immune modulatory drugs. But I think it needs time to test that.
Yeah, of course. Well, thank you very much for that. I think we are out of time for any more questions, but if there are questions, please feel free to pop them in the chat. We do have some time later on to address them. So as a reminder, throughout the session, please keep those questions coming. So thank you again, Professor Kataoka, for a really great talk. And now we'll move on to our next speaker, who is Associate Professor Jane Oliaro. So Jane is a group leader in the Center for Cancer Immunotherapy and Chief Scientist for the Center of Excellence in Cellular Immunotherapy Translation Laboratory at the Peter MacCallum Cancer Centre in Melbourne. So Jane completed her Bachelor of Honours degree at Monash University in Melbourne. She went on to do her PhD also at Massey University in New Zealand, and followed by postdoctoral training in France at INSERM Montpellier, and also Australia at the Peter Mac. So truly an international career. Jane's research program uses a range of immune-based assays, genetic screening, and preclinical models to dissect the complex interplay between the immune system and cancer, with a particular focus on tumor immune evasion and resistance to immunotherapy such as checkpoint blockade and CAR T-cell therapy. She also leads a preclinical program focused on the development of novel cell-based immunotherapies for translation into pilot clinical trials through the Center of Excellence in Cellular Immunotherapy. So Jane's talk today is titled CRISPR Screening to Elucidate Mechanisms of Tumor Immune Evasion. So thank you so much for joining us, Associate Professor Oliaro.
Thanks very much. I'll just start sharing my screen. Okay, does that look fine to everyone?
Looks great, yes.
Okay, great, thanks. So thanks for the opportunity to present just a little bit of our recent research. So you've heard earlier how single cell analysis can really help us understand response to immunotherapy. I'm going to change tact a little bit here and just provide a bit of a snapshot of some of our studies using CRISPR screening as a tool to identify the genes and pathways involved in resistance to immunotherapy, in order to identify new therapeutic targets for combination approaches. So Professor Park has already introduced you to this famous illustration of the cancer immunity cycle that was published by Daniel Chen and Ira Melman back in 2013, where antigen-presenting cells take up tumour antigens and present these to naive T cells in our lymph nodes, which results in the clonal expansion of antigen-specific cytotoxic T cells that then traffic to the tumour site, recognise the cancer cells and eliminate them. And while this image really highlights a role for CD8 cytotoxic T cells, the recognition and killing of cancer cells can also be achieved by cells of the innate immune system, such as natural killer cells, and adoptively transferred cells, such as CAR T cells, which of course are genetically engineered to recognise a particular tumour antigen. And while immunotherapy approaches are essentially designed to either boost or mimic this cancer immunity cycle. And while immunotherapy, as you've heard, has proven remarkably successful in some patients, particularly those with metastatic melanoma and advanced lung cancer, as with most therapies, there are ongoing challenges. And there have been now thousands of patients treated with immunotherapy, but optimal responses are still only seen in around 20 to 50% of patients, depending on the therapy regime and the disease. And so determining how we can predict which patients will respond to immunotherapy is, of course, as you've heard, a major area of research and can relate to numerous factors such as tumour size, neoantigen load, and immune cell infiltrate into the tumour, which of course can be revealed really beautifully by single cell analyses. But it also comes down to tumour immune resistance. And so identifying resistance mechanisms is one of the areas we've been particularly interested in, and we've approached this using CRISPR screening. So to address the question of how tumour cells evade immune cell detection and killing, we've been conducting whole genome CRISPR-Cas9 knockout or loss of function screens, both in vitro and in vivo. So this is a schematic of a typical in vitro screen where we use tumour cells that are engineered to express this enzyme, Cas9, shown in green, which is a DNA endonuclease that has two active sites that each cleave one strand of a double-stranded DNA molecule. So as many of you know, to delete a gene using CRISPR-Cas9, this enzyme is guided to the target DNA by an RNA molecule or guide RNA that contains a sequence that matches the sequence to be cleaved. And so RNA-guided Cas9 activity then creates site-specific double-stranded DNA breaks, which can then be repaired by non-homologous end joining. And this gives rise to small indels and a loss of function mutation within the target gene. So these tumour cells are lentivirally transduced with libraries of guide RNAs that target all of the genes in the mouse or human genome, such that each tumour cell contains a single gene knockout. We then expose these tumour cells to the relevant immune effector cells and grow out the resistant tumour cell pool and use Illumina sequencing to identify the enriched guides and the genes they target. And we've been interested in looking at resistance to a range of effector cells such as CD8 T cells, NK cells, and CAR T cells. So this is the results of one screen for tumour genes that confer resistance to T cell killing, shown here in this magic plot, in which the top scoring guides and the genes they target are ranked based on their log 10 p-value. So this data shows that deletion of genes such as caspase 8, JAK1 and beta-2M, for example, all conferred resistance to T cell killing. And we find using string analysis that many of the top scoring genes cluster into the interferon signalling and the antigen presentation pathways. And this is a finding that's nicely supported by a series of papers that have identified loss of function mutations in genes in these pathways in patients that have become resistant or are resistant to checkpoint blockade immunotherapy, for example. But perhaps more surprising was that our data strongly suggested that tumour cells evade T cell killing by activating the cytotoxic potential of TNF. And this model depicts why we think this. So upon antigen recognition, T cells will kill via the perforin granzyme pathway, but at the same time release large amounts of interferon and TNF. And so while interferon can act to upregulate MHC class 1 and further promote T cell recognition of antigen, we think TNF might be even more important because it can bind to bystander tumour cells that might be negative for that antigen and kill via the extrinsic apoptotic pathway, which is really important when you're looking at the heterogeneous nature of tumours. And so loss of any of these, if any genes involved in these pathways, can facilitate immune escape. So this led us to investigate the potential of SMAC mimetics to improve response rates to immunotherapy, as this class of drugs can sensitise cancer cells to TNF-mediated cell death. And we were particularly interested in determining whether the SMAC-mimetic birinapant would enhance CAR T-cell efficacy for solid tumours, because we know CAR T-cells produce large amounts of TNF. So we first treated mice bearing HER2-positive tumours with anti-HER2 CAR T-cells in combination with birinapant. And so this graph shows the tumour growth with the untreated control in black. So while CAR T-cells or birinapant had a modest effect on tumour growth, the combination therapy significantly controlled the growth of these tumours, with over 50% of the mice being cured. So to then test if this combination therapy might be effective in human cancer, we also investigated this combination in the context of colorectal cancer tumoroids, which are grown in matrigel from patient biopsies. And given many of these tumours actually express HER2, as shown on the plot on the left there. So for this, we generated human anti-HER2 CAR T cells, and we tested these on this patient's tumoroids using red fluorescence as a marker of cell death. And using this model, we demonstrated that CAR T cells alone were able to recognise and kill the tumoroids, and that the addition of birinapant alone had a modest effect. But again, in combination with CAR T cells, the addition of birinapant caused significant tumour cell death that we demonstrated was dependent on TNF. So we're also interested in identifying tumour genes that confer sensitivity to NK cell killing when deleted. So we performed whole genome CRISPR-Cas9 sensitivity screen with NK cells as the immune pressure. And the results are shown in this magic plot on the left, and showed that deletion of genes such as JAK1 and 2 associated with the interferon signalling and TAP2 and beta-2M associated with MHC class 1 expression rendered the tumour cells most sensitive to killing by NK cells. And this, of course, makes sense if you consider one of the key recognition modes of NK cells, where on healthy cells, MHC class 1 binds to inhibitory receptors on the NK cells and prevents them from killing healthy non-malignant cells. However, when a malignant cell loses expression of MHC class 1, this allows the NK cells to recognise and kill the cell, which is a phenomenon we all know as missing self. So given the known association between loss of interferon signalling and resistance to checkpoint blockade immunotherapy, we next investigated the connection between T-cell resistance and NK cell sensitivity using these unbiased CRISPR screens. And so to do this, we compared the enriched genes from our T-cell resistance screen that I showed earlier with the depleted genes from the NK cell sensitivity screen. And we found this common set of genes that when deleted confers resistance to T-cell killing, but sensitivity to NK cell killing. And so together, these two studies demonstrated that loss of function gene mutations in the interferon pathway confers resistance to CD8 T cells, but sensitivity to NK cells. And given that loss of function gene mutations have been in this pathway been identified in patients resistant to immunotherapy such as checkpoint blockade, this suggests that NK cells may present a potential immunotherapeutic strategy to overcome and limit T-cell associated tumour immune resistance, either by adoptively transferred NK cells or through therapies that enhance NK cell activation and effector function. Finally, we noticed in that last screen that as well as genes involved in antigen presentation and interferon signalling, these two genes here, RNF31 and RBCK1, conferred sensitivity to NK cell killing. And by comparing our NK cell screen data with published data that had screened for sensitivity to T cell killing, we found again a core set of genes shown here that confers sensitivity to both effector cells. So RNF31 encodes for HOIL1 interacting protein, or HOIP, which is the catalytic subunit of the linear ubiquitin chain assembly complex known as LUBAC. And LUBAC we know is needed for stabilization of TNF receptor 1 signaling and is required for TNF-mediated gene induction and to prevent TNF-mediated cell death. And indeed, we were able to demonstrate that HOIP-deficient B16 tumours were indeed more sensitive to T-cell mediated control. And this significantly enhanced survival of mice, suggesting that HOIP activity may be used to sensitise tumours to immunotherapy. We also wanted to determine if we could phenocopy genetic deletion of HOIP with a pharmacological inhibitor using a recent HOIP inhibitor called HOIPin1. And strikingly, what we found was pre-treatment with HOIPin1, whilst not intrinsically toxic to the tumour cells, significantly sensitised what would otherwise have been resistant parental B16 melanoma tumour cells to cell death induced by combined TNF and interferon, or to supernatant from NK cell tumour cell co-cultures. And importantly, this was also observed for a human melanoma cell line shown here on the right. So while I haven't shown you all the data, this latest study demonstrates that HOIP limits tumour sensitivity to both NK cell and CD8 T cell cytotoxicity in that it protects against apoptosis induced by combined TNF and interferon. And we've also demonstrated that pharmacological HOIP inhibition sensitizes tumour cells to immune cell-derived cytokines. And so overall, this suggests that HOIP inhibition may be a potential strategy to enhance immunotherapy responses. And this is something that we're interested in further investigating. So in conclusion, I think this brief snapshot of these studies has demonstrated the power of CRISPR screening to identify genes and pathways important for immunotherapy responses, and has helped identify new targets to improve immunotherapy responses. And our data suggests that TNF is a key effector mechanism that can be harnessed to boost tumour cell death using SMAC mimetics, that boosting NK cell activity may overcome T cell mediated immunotherapies, resistance to T cell mediated immunotherapies, and finally, targeting Lübeck complex may increase sensitivity to immune cell derived cytokines. And I think I'm just about out of time, so I'll end with this slide, thanking all of the people involved and the funding bodies on the right.
Thank you so much, Jen. That was such an interesting talk, and right on time, and you're finished. So thank you for sharing it with us, that really exciting research. I think we have time for just one quick question. You highlighted the subset of tumor genes that you found that you said confer sensitivity for NK killing. I'm curious to know, have you done more work into understanding what might be the network of how these genes are connected to each other, or how the robustness behind their ability to give these cells that property?
So what these CRISPR screens will identify is those dominant pathways. So we know that all of those genes that we hit in our screens all fall into some part of the either interferon or TNF signaling pathways. And of course, those pathways are very well identified now. But what they can also do is reveal some of the novel regulators of those pathways. And in terms of like the latest data on the Lübeck complex, it's actually fairly well characterized how the Lübeck complex works in terms of determining sensitivity to TNF mediated signaling. So I guess in a way, a lot of these studies highlight what we know about effector function, but the beauty is, because they're completely unbiased, is that they also can reveal novel targets. And yeah, we're trying to figure out how some of those novel targets fit into the genetic pathways that we uncover during these screens.
Yeah, no, that sounds really cool. Okay, so I think in the interest of time, we might move on to our next speaker, but thank you again, Jane, for a really fascinating talk. And I'll hand over to my co-chair to introduce the next speaker.
Okay, hi, I'm gonna share the other 4 speakers for this session. I'd welcome the speaker from Korea, actually, Murim Choi. He's a professor in Cellular University Medical School, and he was trained in the Cellular University, also at Duke, and postdoc training in Yale. And he studied rare disease, also many other human diseases with the bioinformatic tools. So he now is focused on the screening of the eQTL signals in only function in the non-alcoholic fatty liver diseases. So today he's gonna present us about the screening disease-specific eQTLs using single nuclear RNA-seq of human liver tissues. Please welcome Dr. Murim Choi.
Thank you, Woong-Yang, for the nice introduction and the opportunity. Let me start sharing the screen. All right, so I'd like to show you our ongoing project on the screening disease-specific eQTLs using single nuclear RNA-seq of human livers. Our disease model is a non-alcoholic fatty liver disease, and as you know, it's a very typical common complex trait which affects about quarter of adults worldwide. Many risk factors are known, and there is 22 to 50% of heritability is known as well. So what genes are involved in the conferring the risk or protection of this disease? To answer that, people has done some GWAS studies, but as you can see from here, from several Manhattan plots, compared to other major chronic diseases, the NAFLD GWAS is not that robust. You can still appreciate the very outstanding signal TNPLA3, but other than that, the genetic contribution seems to be very low. And along this, there has been very interest in developing the drug against NAFLD, but as far as I know, many has failed so far. The reason is probably common to many different common diseases, but the heterogeneity in the NAFLD patient is quite severe. And people also think that the selection of the right participant is a key to efficient and successful clinical trial. So hopefully trying to, rather than performing the GWAS, but hopefully trying to reduce the dependency on sample numbers, and also by conferring the patient-specific effect to the understanding of NAFLD pathogenesis, we decide to perform the eQTL analysis. First, the Sung-eun, my graduate student in our lab, is performing the most of the single cell analysis. And Tae-kyung has been, I'm going to show you a little bit of a previous study on bulk RNA-seq data on NAFLD by Tae-kyung. And Professor Won Kim, who is the main provider of NAFLD needle biopsy samples and clinical information as well. And also Professor Kyung-Seok Seo, he has provided us some very precious control liver tissues from the liver transplantation. So they are both in Seoul National University hospitals. And the company Genius has produced a very nice quality of single nuclear RNA-seq data. So let me first introduce a little bit of our previous response eQTL analysis on bulk RNA-seq data. So using Professor Won Kim's samples, we recruited 83 NAFLD livers and 42 non-NAFLD or healthy livers. And we generated bulk RNA-seq data. And from the same participants, we used their DNA, blood to extract DNA and type their SNPs. And we end up by getting 3 million of SNPs. So by combining those 20,000 transcripts and 3 million SNPs, you can perform the eQTL callings. And you can, using the 125 individuals, you can do the liver eQTL callings, which most of the studies so far has been doing. But we thought that this approach, although this gives you a very nice overview of liver biology in general, this does not give you much about the clinical implication of each liver eQTLs. So we decide to divide them, the cohort, into controls and cases, and separately called eQTLs. And we selected the eQTL signals that are only present in the NAFLD group. So we call them as NAFLD eQTL. By doing it, we hope that we would be able to identify the eQTL signals that are only active in the NAFLD context, cellular context. So these are the examples of liver eQTLs and NAFLD eQTLs. As you change genotype in NAFLD group and non-NAFLD group and in GTEx, those liver eQTLs behave in the same manner, which is very typical of eQTLs. But what we were interested in are more like this. So they change, the expression changes by genotype only in NAFLD group, but not in the non-NAFLD or GTEx group. So we set out to systematically call them and we devised a bioinformatic pipeline to end up with calling 243 of such eQTL signals from the livers. This plot shows you the scatter plot of Bera value of a nephrotic group and non-nephrotic group, and shows you that the dark blue ones, which we finally think that the nephrotic eQTL shows a quite high absolute value of Bera nephrotic values, but compared to that, very low minimal value in absolute value of non-nephrotic Bera values. Among those, we set out to select the gene called AGXT2 for the functional analysis, and which includes an alanine glyoxylate aminotransferase 2. And again, if you look at this eQTL plot, the SNP RS22917002 as the genotype of that SNP changes from TT to CT to CC, the expression level of AGXT2 drops. But this is not happening in the control group or GTEx group. This gives you quite an interesting clinical implication, because if you can assume that you can develop a drug against AGXT2 and somehow you can increase the activity of it, then you don't have to prescribe drug to everyone. You can only select the one that has CC carriers, and then prescribe drug on them. So this gives you the quite good resolution on the patient selection. And to just going on to the other data, we were able to replicate this result using 170 additional nephrotic patients. And not only showing this by RNA level, we tried to confirm this in the protein level by performing immunohistochemistry of AGXT2 protein. And then multiple rounds of immunohistochemistry and quantification of the signal showed that the difference in the protein level is also happening in the nephrotic group, but not in the control groups. After subsequent experiments, which I'm not showing you today, we were able to observe that by dividing the cohort into cases and controls, and separately code the eQTL individually, and then looking for the specifically active eQTL in the nephrotic group, we were able to identify 243 eQTL signals. Among those, we think that the AGXT2 can be a quite interesting candidate, because it shows you, which I have not shown today, is that using cell models and mouse models, by reducing AGXT2, we were able to recapitulate fibrosis model, human nephrotic model by increasing fibrosis. But conversely, by increasing AGXT2 level in the high-fat diet liver, we were able to rescue it. So, and then as the coming flow of single cell sequencing, obviously our next target was, what if you can repeat that analysis on the single cell level? So we wanted to extend it. So basically we collected additional samples for RNA-seq, and we generated data and performed QA/QC and expression analysis and eQTL analysis, and then response eQTL analysis. So this is our goal. And today, since this is an ongoing project, I think I can only show you the result of a little bit of an eQTL analysis part. Before starting the study, we first performed a simulation and trying to get some idea of how many samples do we need and how many cases and controls do we need. So based on the early frequency, we were able to see that if you collect 50 individuals, and if you have on half, like 0.5, half of one cases and half controls, you can achieve little less than 90% of sensitivity of SNPs, common variant. So we set out to collect those samples using the collaborating with clinicians in the hospital. We recruited 48 tissues, and among those, 25 are affected and 23 are unaffected. And among the affected, 6 were NAFL, and 9 was early NASH, and 10 were advanced NASH. And among the 23 controls, 20, most of them came from the surgery department, which are chunks of tissues from the transplantation. And the 3 came from the needle biopsy from the internal medicine. So out of 48 tissues, we were able to get the nice data of 249,000 of nucleus. So this is the overall UMAP of 250,000 of cells. You can see that the major cells, including the non-immune cells, are quite well represented, and they were also very well distributed through different classes of the tissue samples. And by looking at the brief overview of sample composition by the classes of clinical status, you can appreciate that probably the most notable thing is that as you go from the normal to NAFL and early to advanced NASH, there is a higher contribution of immune cells at the expense of less contribution of hepatocytes. And then we perform the pseudotime analysis on the major cell types and trying to see whether they recapitulate the known cellular status. So hepatocytes, if you are familiar to the liver structure, the liver is composed of very small hexagonal structures called as a lobule. And each lobule is, there's a center, there's a vein, and then there is a peripheral region as well. So there is always a central to peripheral axis in each lobule. And then previous study shown that in using the mouse liver, they have shown that single cell analysis can efficiently localize the location of the cell by looking at the peripheral or pericentral gene expression. So we performed the pseudotime, and first we clustered the hepatocyte only, and then we identified 2 different lineages. And the first lineage especially was showing the peripheral to pericentral gradient quite nicely. So peripheral genes was mostly reduced by the ongoing pseudotime, whereas the pericentral genes was increased by ongoing pseudotime. And by simply comparing the genes between the cases and controls, you were able to see that the NAFLD NAFLD cells were enriched with an acute phase response and blood microparticles, whereas they were reduced, the response to insulin or oxidoreductase activity genes were reduced in the NAFLD group. And stellated cells, we were also identify 2 lineages, and one was going from quiescent activated, and the lineage 2 was going from quiescent activated to inactivated, which was also recapitulated by the quiescent markers and activated markers, and so on. And the gene expression change showed you that in the NAFLD group cells, extracellular matrix and GTPase regulator and epithelial to mesenchymal transition genes were enriched in the NAFLD group. Cholangiocytes, we were able to see the progenitor to differentiate the lineages, and again, nicely shown by each markers. And the gene expression analysis showed you that there is a cytoskeletal protein binding, and GTPase regulator genes were unregulated in the NAFLD, whereas the structural constituent of ribosome genes were reduced in the NAFLD group.
Last 20 seconds left.
Okay. Last, for the endothelial cells, we were able to see the pericentral to peripheral gene axis, and there is an increased expression of angiogenesis and VEGF signals. Cell phone DB analysis showed you that there is a difference in the interaction between 2 different subsets. Among those, we think that the macrophage and the stellate cell or monocyte interaction was quite notable. And we were also performing eQTLs, which is quite similar to, we are just showing the examples of an eQTL, which is quite similar to GTEx examples. And also, we are also performing the further analysis on the eQTL by dividing them into the different detection from the different tissues. So trying to sum up this talk, we trying to extend our previous bulk eQTL studies and trying to devise a single cell level response eQTL using human liver samples of various clinical status. And our data demonstrate the major cellular features for tissue-specific diseases. And I showed you that there is quite an interesting, we were able to detect a quite interesting gradient of a cellular status. And also we are actively pushing on the tissue-specific eQTL and response eQTL analysis. So hopefully we can show you the interesting result in the later opportunity. Thank you.
Thank you. Thank you, Murim. So maybe a little bit late, so we're gonna skip the Q&A session. But at the end of our session, we can ask the question from the floors in the later. Thank you. Please stay in the session. Thank you. So next speaker from Chinese University of Hong Kong, Yingying Wei, Dr. Yingying Wei. So she studied in the mathematics and also in the computer science in her training in the Hopkins. And also she developed many interesting methods with the batch correction method. So she awarded from the American and Statistical Association in 2019. So she will present us today about the differential inference for the single cell sequencing data. Please welcome Dr. Wei.
Yeah, thank you very much, Professor Park, for the kind introduction. So can you see my screen now?
Yes.
Slide? Okay,
yes.
Thanks so much. Yeah. So first, I want to thank the organizers for this precious opportunity. So today I will talk about differential inference for single-cell RNA-seq data. So this is joint work with my former student, Fang-Na Song. So at the end of, the single-cell RNA-seq experiments, usually we can get a set of matrices like this. So where each row corresponds to a gene and each column corresponds to a cell. And usually we will have multiple batches of data. So each batch can consist of samples, cells collected from multiple individuals, for example. So how to correct the batch effects? So as if all the samples were measured in one single batch is key problems in the past several years computational biology. So please bear with me before I go to the differential inference for single-cell RNA-seq data. So I want to briefly discuss a little bit about the batch effects correction for single-cell RNA-seq data. So the main challenges is that how can we correct for the technical artifacts, especially when we have the unknown biological variability? Because one of our main goals is to do the cell type clustering, but we don't know the cell types for each individual cell. So we have both the biological heritability as well as heterogeneity, as well as the technical heterogeneity. And meanwhile, we also encounter dropout events in single-cell RNA-seq data, which means a gene is actually expressed, but we fail to detect that in our data. And the data are count data, so we have the common overdispersion issue with the count data, as well as for each individual cell, we will have the cell-specific sequencing depths. So the commonly used popular batch effects correction method for single-cell RNA-seq data include the MNN, Scanaroma, and SERRAT. So SERRAT later versions also borrows ideas from the MNN. However, MNN actually relies on two assumptions. So the first assumption is that the batch effects variation is almost orthogonal to the biological subspace. And the second assumption is the batch effect variation is way smaller than the biological effect variation. Unfortunately, when we look at the real data, so both assumptions may not hold. For example, so these are two data sets from the MNN papers. And if we look into the cell type effects as well as the batch effects, so in the heat map, each row corresponds to a gene and each column corresponds to a cell type or a batch. So as we can see, both the magnitude of the batch effects can be quite substantial. And also they are not orthogonal, right, to the cell type effects. So therefore, we proposed a method that called batch effects correction with unknown subtypes for single-cell RNA-seq data, BUS-seq. So that is a hierarchical model, so that can try to simultaneously correct batch effects, cluster cell types, and take account of the overdispersion, the dropout events, and the cell-specific sequencing depths. So there's no need to do cell-specific normalization. So later, our differential inference were based on the BuzzSeq. So please allow me to use some time to introduce the model. So the idea is to say for the ice cell measured on the B's batch, so its probability to belong to the K cell type is pi BK. So then given the cell belongs to the K cell type, so then its underlying gene expression level for the G's genes will follow an active binomial distribution with a mean of mu BIG and an over-dispersion parameter phi BG. And then the mean on the log scale, so that can be decomposed into the baseline gene expression level, alpha G, as well as the cell type specific effects, right? For example, suppose the second cell type compared to the first cell type, right? So the expression level is 2 units higher on the log scale. So then beta GK equals to 2. And then we also have a gene-specific batch effects, that is nu BG. And finally, we also incorporate a term, delta BI, to characterize the cell-specific amplification factor and sequencing depths. But unfortunately, we are not always able to observe these underlying gene expression levels. So because we have the dropout events, where we use a CBIG to do the indication. And then the dropout rate, we use a further logistic regression models. And as we know in the single cell RNA-seq data, so the dropout rate will actually depend on the underlying gene expression levels. So the more highly expressed genes are less likely to suffer from dropout events. And then finally, suppose it is a dropout event, so then our observed data will be 0. And suppose it is a successful detection, so then we are able to observe the underlying gene expression levels. But unfortunately, right, so our observed data only correspond to the gray box, while the yellow boxes are all latent. But fortunately in statistics, so we have techniques that we are able to learn all these latent variables and estimate these parameters well. So we developed an MCMC algorithms to do the statistical inference. And when applied to the data sets, so that is the one from the MN, as we can see, our method actually outperforms those state-of-the-art method by then in terms of both ARI and the salute coefficient. So it's an MCMC algorithm, so we provide both the parallel C++ code, the parallel CPU code, and also the parallel GPU version, the CUDA-C GPU version for our algorithm. So you are more than welcome to have a try. So now let me move on to the main issue we want to discuss today, right? So that is how we can do the differential inference for single cell RNA-seq data. So that is now we want to compare the single cell RNA-seq data collected from different biological conditions or under perturbations. So this actually, there are emergent works that try to quantify the treatment effects for single cell RNA-seq data. For example, the MELD. So their main target is try to quantify, right? So the change in terms of the enrichment of the cellular compositions between the experimental perturbations. And however, they need to assume the gene expression levels of cells in the same state pretty much keeps us invariant under different conditions. However, as we can expect, right? So under different biological conditions or perturbations, not only the cellular compositions may change, but the underlying gene expression levels may also change. There are also recently proposed methods that try to model how the gene expression levels will depend on the cell types and the perturbations and other covariates using deep learning-based techniques. But however, so the rigorous statistical testing approaches to quantify the uncertainties are still lacking. So the main challenge is, of course, right? So you can do multi-stage approaches. For example, you can first try to do cell-specific normalization, then try to correct for the batch effects. After the batch effects are corrected, then you try to cluster the cells into cell types, and then you try to impute the missing data caused by the dropout events. And then based on, after the cell types are clustered, then you try to identify those differentially expressed genes. But unfortunately, right, so each step can be prone to errors, and the errors can be propagated to the further steps. And moreover, when you do the cell type clustering, if you don't consider those differentially expressed genes, you may also run into trouble. So our target is we try to propose rigorous statistical inference approaches that can detect the changes in the cellular compositions as well as the changes in the cell type specific gene expression levels. So we build upon our previous model. So we only actually add one extra term, right? So that will that is eta TGK that try to quantify the treatment effect. But however, statistically, there are many things we need to do because in the past, we only care about the point estimator. So now we want to quantify the uncertainties. So we conduct a statistical inference under the Bayesian framework, and we use a spike and slab pliers to do the selection, whether it's a 0 or not. And then instead of the MC algorithm we used in the past, so now we use an expectation conditional maximization algorithm with stochastic updates. So we can handle very large data sets, like consist of over 200,000 cells. And we use the Lewis estimator to quantify the uncertainties and use BIC to determine the cell type number. So our proposed method is able to detect those intrinsic genes that are those genes that are differentially expressed between cell types. And also, we are able to detect those genes that are differentially expressed between treatments or perturbations or biological conditions and in a cell type specific manner. And also, we are able to detect those differences in the cellular compositions. So in statistics, we always do simulation studies to verify the method. So here I just to show a very simple simulation study for the convenience of discussion. So we have 2 batches. On both batches, both treatments are measured. So then there are a total of 4 cell types. And then for each batch, right? So you can see a batch 1, so 1, and 2, so they correspond to the same treatment. And 1, 2, and 1, 1, they correspond to 2 different treatments, but they are measured on the same batch. So then our BIC is able to successfully detect that there exist 4 cell types in the data set. And we are able to recover the true underlying parameters. So this is for the cell type effects. So for the left-hand side, so that's the log scale mean expression levels for the 4 cell types. And the right-hand side are the estimated ones. And for the treatment effects, right, so we are also able to detect them quite successfully. So the first row of the color bar corresponds to the treatments, and the second color bar corresponds to the cell types. So we have cell type specific treatment effects. And also for the batch effects, so they're also gene specific. So we have 2 batches, so we're also able to recover that. And we're also able to estimate the cell specific amplification factors pretty accurately. And we are, for the comparison of the cellular compositions, so we can do both a global test to see, okay, whether these 2 sets of cellular compositions are equal or not. So we can see between these 2 conditions, between the 2 treatments measured on the same batch, we can see actually we reject the null hypothesis. But meanwhile, for the 2 treatments measured on 2 different batches, so we fail to reject the null hypothesis, right? Because their cellular compositions are the same. And meanwhile, we are also able to do element-wise specific test. So that is, we are able to detect, okay, so which cell type is actually the composition will change between conditions. For example, in our simulation setting, as we can see, so the cell type 1 and cell type 2, their proportions stay the same across conditions, but the proportion of cell type 3 and 4 will change, right? So this is actually successfully detected by our method, right? So for the cell type 3, right, so we reject the null hypothesis. And our method actually achieves better clustering results. So the ARI is 1 for our method in comparison, so the SIRET is only 0.43. So we also apply our method to the previously discussed integrated pancreas studies, but this time we'll also consider the condition of the patients where the cell came from. So some cells are collected from the healthy individuals, while some cells were collected from type 2 diabetes patients. So as we can see, once we consider the biological conditions of the cells, so actually we can further improve our ARI. In the past, our methods was 0.61, but now we can improve to 0.82. And meanwhile, so we are able to detect those intrinsic genes that are differentially expressed between cell types. And our method detected 384, while by SIRET, so they pretty much call all the genes as differentially expressed between cell types. So for the cell type specific differentially expressed genes, as we know for diabetes, right? So the beta cells are really important. So we can see those 9 genes identified by our methods are actually pretty interesting. So as a summary, so we proposed a method that is called BUT-seq. So the purpose is we want to do a rigorous statistical test to detect those intrinsic genes, and that are the genes that are differentially expressed between cell types. And also we want to call cell type specific differentially expressed genes, because we can expect the biological conditions may change the gene expression in some cell types, but not in all cell types. And we want to provide statistical tests to test whether there's any difference in the cellular compositions between conditions. Meanwhile, our method can automatically correct the batch effects plus the cell types into the missing data, and there's no need to do any normalization. So I want to thank Fonda for working on this project, and Angus for working on the BUT-seq and the funding support. So thank you very much for your attention.
Thank you, Dr. Wei. Also, we are a little bit behind the schedule, so we're gonna skip the Q&A session, Q&A, but actually we're gonna ask other questions from the floors in the end of this session. Please wait, Dr. Wei. Thank you. And then next speaker for this session be from Singapore, GIS, Singapore Genome Institute of Singapore, and that Dr. Ignasius Joanito. He was born in Indonesia and then trained in Taiwan and China, and now working in Singapore. And actually, he studied in the Genome Institute of Singapore, and interesting about analyzing single-cell RNA-seq data in the colon cancer. So he developed a many methods for the mathematical modeling to understand dynamics. So he will present us today about single-cell and bulk transcriptomics review 2 epithelial cancer subtypes in colorectal cancer. Please, Dr. Joanito.
Thank you, Dr. Park, and thank you also to the organizer for this opportunity. My talk will be about new scRNA-seq based classification of colorectal cancer. So, as you may already know, colorectal cancer tumor is very heterogeneous, such that each tumor is different. As a result, a lot of effort has been done to develop a good classification method to stratify this patient into bins, so we can treat patient in each group separately. At least that is the dream. Currently, the molecular consensus subtype, or what we usually call as CMS, is the currently state-of-the-art transcriptomic classification for colorectal cancer. It classifies CRC into 4 different groups: CMS1, CMS2, CMS3, and CMS4. Actually, CMS4 is the one that characterized by the stromal infiltration and TGR beta activation, and also angiogenesis is the most metastatic subtype. The limitation of the CMS classification is that it is still based on the bulk scRNA-seq data, which will obscure the signature of distinct cell population and microenvironment interaction. Since then, a lot of effort has been done to address this limitation. For example, in 2017, Isela et al. Proposed a new classification system called CRISPR classification. Actually, in here, the expression of human tumor is propagated as patient-derived xenograft, where human stroma cells are experimentally depleted. However, this xenograft approach is limited by the possibility of artifact from the propagation in mouse. So, to construct a more high-resolution classification, we perform scRNA-seq on primary human tumor for multiple cohorts. Here, we collected data from 5 cohorts and from 3 different countries, which we call as CRC-SG1, CRC-SG2, CAL3, CAL5, and SMC. In total, we have 188 samples from 63 patients, which generated around 421,000 high-quality cells. In this study, we also will focus our analysis on the epithelial cell, which is to the best of our knowledge, is the one of the largest CRC data set today. After that, we also validate our finding using the 3,614 bulk transcriptomic data from 15 cohorts before finally perform an integrative analysis. For epithelial subtype, we found that most of the cells are forming a patient-specific cluster. However, we believe that it is not due to the batch effect because epithelial cells from the nearby normal were still clumped together and form a single normal cluster. Next, we calculated a pseudobulk transcriptome from this patient-specific cluster, and using PCR analysis, we found that two separate groups are self-emerged with a very distinct marker, as you can see in this figure here. After that, we validated this finding using 4 independent cohorts, the CRC-SG2, SMC, CAR3, and CAR5. Here, we use all cells using we cluster all cells using the D gene that we found in the previous step. As you can see in here, we again found that most of the epithelial cells from a patient-specific cluster, and there is still a good separation between the normal cell from the tumor cell. Mostly the normal cells still form a separate cluster, and it is it's not clustered together with each patient separately. Using the PCR analysis of patient-specific pseudobulk, we again found 2 groups are self-emerged in this 4 cohort, as you can see in this figure here. Actually, this finding is also support our previous observation in our CRC-SG1 cohort. In fact, with this finding, we perform another independent analysis using Cynic pipeline and calculate the regulatory activity of transcriptional factor and also their target gene, or what usually called as the regulons. We cluster our cells from 5 different cohorts using the 362 regulons that we found, and again we found 2 distinct groups are self-emerged in this clustering analysis. Furthermore, we also found that 64 differentially expressed regulons from these 2 groups, including some very interesting regulons ATF, such as TCF7, MYC, and also some of the NF-kappa Bs, defining these 2 group features. Previously, CRC tumor has always divided into 2 groups. The first one is the microsatellite stable, or the MSS group, and then the second one is the microsatellite unstable, or the MSI group. Recently, there are many interest in the MSI tumor because it shows better response to immunotherapy treatment, as you probably already hear in the talk, Dr. Park talk in this morning. And then in this study, we found that all MSI tumors are found within the I3 subtype. However, we also found that some MSS are clustering together with the MSI tumor, suggesting biological program in the I3 MSS, which are more similar to MSI high than the I2 MSS. The dendrogram construction based on the transcriptomics distance also pointed to distinct differences between the 2 intrinsic subtypes, with much greater similarity between the I3 MSS with the I3 MSI compared to the I3 MSS versus I2 MSS. Actually, this result is very interesting because previously people always assume that there is a high level dichotomy between the MSS tumor and the MSI tumor. Hence, understanding the immunological makeup of the I2 MSS and I3 MSS cancer could inform subtype-directed development of immunotherapy treatment. Next, we try to validate this finding in the bigger cohort using the bulk RNA-seq data. Here, we use our single-cell ICMS marker to classify the 3,614 tumor bulk transcriptome from 15 datasets. And similarly, observed that there are two groups of patients, one with higher expression in ICMS2 signature, while the other has a higher expression of I3 CMS signature. After that, we examined the relationship of the ICMS subtype with associated clinical and molecular features across the bulk dataset. Here, we found that most of the I2 tumor are left-sided, while most of the I3 tumor are more right-sided. All MSI are still within the I3 tumor. However, we again see that some of the MSS tumor are co-clustered with the I3, as we also observed in our single-cell data. CMS1, CMS3 are mostly I3, while CMS2 are mostly I2. However, we also found that CMS4, or the metastatic subtype, can either be I2 or I3 with almost equal proportion, suggesting that the fibrosis process might be separated from the intrinsic epithelial structure. Next, we combine the bulk transcriptome into a single gene expression matrix and perform a de novo clustering on it. Here, we again found a self-evident co-organization by MSI status here, CMS, this line, and also the ICMS here, which support that they might provide the underlying structure to the molecular classification of CRC. Unfortunately, we cannot observe a clear pattern based on the CRISPR classification, as you can see in this figure here. We also observe that most of the MSI tumor are immune infiltrated, which was indicated by high expression level of genes that are expressed in the immune cell, as you can see in this bar chart here. And then also most of the CMS4 are fibrotic, which indicated by high expression of gene that is expressed in the in the fibroblast and the endothelial cell. In this part of the heat map. And again, we found that CMS4 are split into two separate groups depending on their ICMS status, either they are ICMS2 or ICMS3 C4. Which is also might suggest that the epithelial subtype and fibrosis are a decoupled process. Yeah. As reported previously, we also observed bad relapse-free survival of CMS4. And then, however, for the poor RFS and overall survival, actually it was the features of the I3 CMS4 group. This result is very interesting because it indicates that CMS4 cancer with I3 epithelium should be the focus of biological and clinical studies seeking to prevent metastasis in CRC. Based on the previous result, we therefore propose a reorganization of the four group ICMS into the four group of CMS classification into 5 group type of classification based upon 3 biological layers. The first one is intrinsic epithelial status, or the I, the microsatellite status, or the M, and then the presence of the fibrosis of the F. Together, we name this classification as IMF classification, which stratified tumor into 5 different subgroups, which you can see in this figure here. So in summary, in this study, we proposed a new 5-way classification of CRC tumor called as an IMF classification. Using scRNA-seq data, we identified 2 intrinsic cancer subtypes shared across 5 cohorts, which we called as ICMS2 and ICMS3. The intrinsic epithelial dichotomy was also found in the bulk CRC transcriptome, where ICMS3 underlined the epithelium of bulk CMS1 and 3, while ICMS2 underlined the epithelium of bulk CMS2. Intriguingly, we also found 1/3 of the MSS tumor, which we call as ICMS3 MSS, are more similar to MSI high than the other MSS, at least at the epithelial level, which is significant because MSI high is the immunotherapy response subtype. Lastly, CMS4 is evenly split between cancer with the I2 and I3 epithelium, in which fibroblastic ICMS3 have the worst prognostic. These findings suggest that epithelial subtype and fibrosis might be separated process. And with this, I would like to thank my mentor, Dr. Shyam Prabhakar, our international collaboration, Dr. Ian Tan, Dr. Sabin Tejpal, and then also Dr. Woong-Yang Park, as well as multiple local collaboration. All team member and Shyam's group, and also all people that are working for this project. Thank you.
Okay, thank you, Dr. Joanito. We will collect all the questions, and also we will summarize in the end of the session. So please stay in this the session. Thank you. And then last speaker of this session will be a graduate student from National Institute of Biomedical Genomics in India, Mr. Smithavan Roy. He is in training of in the PhD program in the Institute of Biomedical Genomics, and with the in the lab of Professor Arindam Maitra. So he will present us today about characterization of cell state heterogeneity in Gingivo-Buccal oral squamous cell carcinoma. Mr. Roy, please.
So thank you, sir, for the introduction. So, you can see my slides.
Yes.
Okay. So is there any problem here? Any tab is opening here?
Yeah, it's okay. Yeah,
okay.
please.
Okay. So good morning, everyone. So today I'm going to present a part of our ongoing research work titled "The Characterization of Cell State Heterogeneity in Gingivo-Buccal oral squamous cell carcinoma." So each tumor is heterogeneous in nature, containing different type of cells, which influences interpatient variability in tumor progression. So, being a cancer of epithelium, oral cancer is predominant in worldwide, with a drastic increase in incidence of cases. And the factors behind the oral cancer are mainly like chewing of tobacco, betel quid, smoking, alcohol, HPV. So in India, OSCC-GB is the most prevalent type of oral cancer due to the habit of chewing of tobacco and tobacco-related products, betel quids, etc. And whereas in Western countries, the tongue is the mostly affected due to the smoking of alcohol, smoking and alcohol consumption. And in India, another factor is that males are mostly affected than the females. Thus, here in our study, we chose OSCC-GB to explore the tumor heterogeneity. The major question that we ask is, what are the cell state heterogeneity in OSCC-GB? So for this, we have profiled cellular diversity in 12 OSCC-GB tumors, which are early stage treatment naive samples collected from Dr. R. Ahmed Dental College and Hospitals, Kolkata. We used a droplet-based 10x genomics platform to generate the single-cell 3' RNA-seq libraries. And these are then sequenced in NovaSeq 6000. The output files are then processed by Cell Ranger and Cellular. So we identified almost 52,000 cells and retained around 25,000 cells after the quality control checks. So we have got an average of around 2,000 cells per sample. So coming to the results, we have got 7 major clusters from the 12 OSCC-GB samples. And these donut plots are showing the proportion of cells of different patients populating each cell type. So basically each cell type has representative cells from all samples. The next, what we did, we divided the cells into the malignant and non-malignant compartments, and we can see that the malignant cells, so these are the clusters that are produced, are majorly clustered according to the tumor of origin, this plot. And if we see in case of the non-malignant cells, so in case of non-malignant cells, they are clustered according to their cell type. So this draws our attention to look deep into the malignant cells, malignant cell clusters. So for this, we did a differential gene expression analysis within the malignant cells subclusters, taking the subpopulation of cells from these malignant cells only to as a reference. And we found pathways that are upregulated in some clusters, whereas in some other the gene signatures are homogeneous. We also found there are some. We also found clusters where the cells from different patients having cancer hallmarks pathways like hypoxia, ACM remodeling, TNF signaling, P53 pathways, cell cycle, angiogenesis, etc. So here we focused on to see the EMT state, epithelial mesenchymal state in OSCC-GB. So briefly, EMT is a major hallmark of cancer that allows cancer cells to acquire mobility and the capacity to migrate. So we saw the expression of EMT markers like SNAI2, SNAI1, ZEB1, ZEB2, and we found that only SNAI2 is present in the cluster 14. And we also look for the mesenchymal and epithelial markers like hymentin and cadherins. So we saw that hymentin and cadherins are also present, though the mesenchymal markers are a bit high and epithelial markers are a bit low. So we can conclude that these cells have the both characteristic of epithelial and mesenchymal cells. And the interesting fact is that though all the markers of EMT is not present in all the malignant cells, only SNAI2 present in the cluster 14. And the previous literature shows that the SNAI2 is expressed early in the EMT process. So we asked a question like, if those cells are not in EMT state, so if there is in a pre-epithelial to mesenchymal state, so for that, we collected the genes from the previous literature that are related to PEMT, and we plotted the average of those genes for all the clusters of the malignant cells. And we found that 6 clusters like 0, 1, 3, 6, 14, 15 had higher expression of for those average PMT genes. And the expression levels are mostly high in the 14 and 15. So next we asked another question that what are the major causal effect cause for these cells to go into the PMT state. So we again go back to the previous literatures and found that hypoxia may play a role in the PMT promoting PMT in malignant cells. So we try to identify if hypoxia-related gene signatures is present in our data set or not. So as you also seen in the previous slide that we got the hypoxia-specific pathways in the malignant cells. So we try to see we see the expression of HIF1 alpha. So we got the expression of HIF1 alpha in some clusters, including cluster 14, but not in all the clusters like 0, 1, 3, 6, where the PMT markers are there. So we plotted the average expression of the hypoxia specific pathways, and again we see that cluster 14 has a higher expression of those genes. So we can say that the cluster 14 was the enriched with cells having high expression of PMT and hypoxia related genes. Next, we investigated the correlation between the average of PMT expression and the hypoxia pathway genes. That again shows the higher correlation in cluster 14. And from here, we identified some genes that may play a role in hypoxia-mediated PMT in OSCC-GB. So from our previous study, we identified ARID2 as one of the driver genes in OSCC-GB. ARID2 is a part of the larger PBAF complex associated to chromatin remodeling. So PBAF complex also already identified to have extensive role in human malignancy. So we investigated the expression of PBAF complex in malignant cells, and we found that not all cells expressing in the PBAF genes, and there are genes that are differentially expressed between PBAF expressing and non-expressing cells. And if we look into gene wise for like added to BRCA and other genes like added to PBRM1, BRD7, they also show the similar results where NEAT1 and MALAT1 are the differentially expressed between PBAP expressing and non-expressing cells, which have the role in earlier reported as a role in recurrence and unfavorable prognosis in other cancers. So slow cycling cells are another aspect that we look into. So these are the subset of malignant cells with higher importance because of its ability of drug resistance, self-renewal, and relapse. So we found the expression of earlier reported marker of SCC, slow cycling cells in the cluster of 14 and 16. And we found genes that are also differentially expressed between these slow cycling and non-slow cycling cells. Okay. So we found the cluster 14 which is a peculiar cluster here because as I have mentioned that the majority of the malignant cell clusters are clustered according to the sample of origin. But there are some clusters like this cluster 14, which are heterogeneous in case of in respect to the sample. So there the cells in the cluster 14 is coming from different samples. So these cells are expressing the PBAP complex genes as well as the we have the expression of earlier reported slow cycling cells marker genes. And there are similarities between the differentially expressed genes between these two characteristics, like NEAT1, MALAT1, PLCG2. We also have the expression of PMT-associated genes within this cluster 14 and hypoxia-associated genes. And we found some genes that may promote the PMT through the hypoxia in OSCC-GB. So to conclude, we can say that OSCC-GB is heterogeneous in respect to cell states, and there is a subpopulation of cells which are common in all the patients having the similar gene expression signatures, and these cells may play a major role in promoting tumor growth, invasion, and recurrence. So these are my collaborators and lab mates. Team that with whom I am working with from National Institute of Biomedical Genomics and Dr. Ahmed Dental College and Hospital. And I want to acknowledge System Medicine Cluster, Government of India, Department of Biotechnology, and University Grant Commission of Government of India. Thank you.
Okay.
So, conclude my.
Thank you, Mr. Roy. And then it's good, nice presentation. Thank you very much. So maybe there's one question from the RBA. He asked, how did you discriminate normal keratinocyte from tumor cells within the tumor samples?
Yeah, so for that we have used Inference CNV and another method that is we have built in our NIBNG in our institute. So that is still in process, and we are trying to communicate those methods in some incoming literatures. So that's how we identified the malignant cells from the epithelial cell proportion from the tumor samples.
Okay. The other question from Shayan BA, to see the dice, to see clear ENT signatures. How prevalent in cluster 14? Also, can you rule out the fibroblastic epithelial doublets?
Yeah, can you repeat one?
Okay. How prevalent is cluster 14? The first question. I mean, how many cluster 14 cells in the frequency of those cells? Or maybe can answer to the second question. Be, can you route fibroblast epithelial doublets for ENT?
Oh, no. So we have checked for those doublets and all things during the quality control checks, and after that only we have processed those cells that are not doublets. We processed only those cells for our subsequent studies.
Okay. So maybe, yeah, time is run. So maybe we're gonna go to the final discussion with the older, inviting the older speakers. We're gonna share with the Jesse together, right?